Hello everyone, this is Farha nausheen of Group 316 and the topic is the structure of nucleic acids. Nucleic acids are polynucleic acids that is long chain like molecules composed of a series of nearly identical building blocks called nucleotides. Each nucleotide consists of a nitrogen containing aromatic base attached to a pentose sugar, which is in turn attached to a phosphate group. Types of nucleic acid. Deoxyribonucleic acid or DNA encodes the information cells need to make proteins, and a related type of nucleic acid called ribonucleic acid RNA comes in different molecular forms that play in multiple cellular roles including protein synthesis. First of all, we will talk about DNA. DNA is known as deoxyribonucleic acid. It is an organic compound that has a unique molecular structure. Functions of DNA. DNA is the genetic material that holds all the inherited information coded in its nitrogen basis structure. DNA passes the genetic information from one cell to its daughters and from one generation to the next. Every person has their own sequence of DNA. This DNA property is used in DNA fingerprinting, a method used to identify a person from their DNA. Structure of RNA. RNA is a ribonucleic acid that helps in the synthesis of proteins in our body. This nucleic acid is responsible for the production of new cells in the human body. It is usually obtained from the DNA molecule. RNA resembles the same as the DNA. The only difference being that it has a single strand, unlike the DNA which has two strands. It consists of only single ribose sugar molecule in it. RNA is also referred to as an enzyme as it helps in the process of chemical reaction in the body. Ribonucleic acid has all the components same to the DNA with only two main differences within it. RNA has the same nitrogenous bases and the adenine, guanine, cytosine for except the thymine replaced by uracil. Functions of RNA. Facilitate the translation of DNA into proteins. Functions as an adapter molecule in protein synthesis. Serves as a messenger between the DNA and ribosomes. They are the carriers of genetic information in the living cells. Promote the ribosomes to choose the right amino acid which is required in building up of new proteins in the body. Types of RNA. The first type is tRNA, the transfer RNA. The transfer RNA is held responsible for choosing the correct protein or the amino acid required by the body, in turn helping the ribosomes. Transfer RNA is located at the end points of each amino acid. This is also called a soluble RNA, and it forms a link between the messenger RNA and the amino acid. The second type, rRNA, ribosomal RNA. rRNA is a component of the ribosome and are located within the cytoplasm of a cell where ribosomes are found. In all living cells, the ribosomal RNA plays a fundamental role in the synthesis and translation of mRNA in proteins. The rRNA is mainly composed of cellular RNA and are the most predominant RNA within the cells of the living beings. The third type is mRNA, messenger RNA. This type of RNA functions by transferring the genetic material into the ribosomes and passes the instructions about the type of proteins required by the body cells. Based on the functions, these types of RNA is called messenger RNA. Therefore, the mRNA plays a vital role in progress of transcription or during the protein synthesis process. And the second topic is translation. The process of translation is a crucial step in protein synthesis, where the genetic code carried by messenger RNA is translated into the specific sequence of amino acids to form a protein. First step, initiation. mRNA binding. The small ribosomal subunit binds to the fifth end of the mRNA molecule. Start codon recognition. The ribosome scans along the mRNA until it finds the start codon AUG, which codes for methionine. tRNA binding. A transfer RNA molecule carrying methionine binds to the start codon via its anticodon region. This forms the indication complex. Ribosome assembly. The large ribosomal subunit then joins the small subunit, completing the ribosome assembly. Second step, elongation. Amino acid tRNA entry. A new tRNA carrying an amino acid enters the ribosome at the A site. Peptide bond formation. The ribosome catalyzes the formation of peptide bond between the amino acid in the P site, peptidyl site, and the amino acid in the A site. This region is facilitated by the ribosomal RNA in the large subunit. Translocation. The ribosome shifts one codon down the mRNA. The tRNA that was in the P site moves to the E site, the exit site, and is released, while the tRNA in the A site moves to the P site. This opens up the A site for a new tRNA. The last step, termination. Stop codon recognition. The elongation continues until a stop codon UAA or UAG or UGA is released on the mRNA. Release factor binding. A release factor binds to the stop codon in the A site, prompting the ribosome to catalyze the addition of a water molecule instead of an amino acid, leading to the release of the newly synthesized polypeptide chain from the tRNA in the P site. Ribosomal disassembly. The ribosomal subunits disassemble and the mRNA and tRNA are released. Fourth step, post-translational modifications. After translation, proteins often undergo various modifications such as phosphorylation, glycosylation, or cleavage, which are essential for their final functional site. Thank you.